Thank you so much for joining us today. This is actually the third explainer in our Mycology series. So if you haven't yet, you might want to check out our Mycology 101 and 201 videos. I've linked them down in the description for you. Today, we are going to untangle a notoriously complex topic in the world of mycology, the many ways we classify fungi. Yeah, if you've spent any time at all in mycology, you know this feeling deep in your bones. Whether you're in a clinic or in a lab, you consult one textbook, then another paper, and all of a sudden you're just drowning in different ways to classify the exact same organism. It can be a huge source of confusion. So today, let's cut through all that noise and find some real clarity. Okay, so before we can even start to classify them, let's just quickly ground ourselves in what makes a fungus, well, a fungus. I mean, we are talking about an entire kingdom of organisms here. They're eukaryotes, so they have a true nucleus. They get their food by absorbing nutrients from their environment. They don't have chlorophyll, so no photosynthesis. And this is the big one, their cell walls are made of chitin. This is our baseline for everything, from a tiny yeast cell to a huge mycelial network. So, how are we going to tackle this beast? We're going to explore three main classification systems. First, we'll look through what I call the clinical lens, which is all about medical relevance. Then, we're going to zoom way out to the evolutionary tree to understand their deep history. After that, we'll get super practical and talk about classifying by simple appearance. And finally, we'll put it all together to help you choose the right toolkit for your specific needs. First up, the clinical lens. Now, this system is all about medical relevance, and it's the go-to for pathologists, infectious disease specialists, and clinical lab scientists. The primary goal here, it's not mapping some ancient evolutionary history. It's all about improving patient outcomes. The key question that drives this entire system is, how is this fungus affecting the human body? So this system organizes fungi based on the diseases they cause. We call these mycoses, and specifically by how deeply they invade our tissues. It's like levels of invasion. It starts with superficial mycoses, which are on the absolute outermost layers, like your skin and hair. Then you have cutaneous infections, which go a little deeper into the epidermis. Subcutaneous mycoses, well, they breach into the tissues right beneath the skin. Then we have the really serious ones, systemic mycoses. These are life-threatening infections that get into our internal organs. And finally, you have the opportunistic mycoses, which, as the name suggests, mainly affect patients with compromised immune systems. And here's what that looks like in the real world. When a doctor sees ringworm, they're immediately thinking, okay, superficial mycosis, probably caused by something like trichophyton. A thorn prick from a rosebush that leads to a nasty lesion, that's a classic subcutaneous case of sporothrix. And for an immunocompromised patient, clinicians are always on high alert for those opportunistic fungi, like aspergillus. See? It's all about context and the clinical picture. Okay, let's take off our clinical glasses and put on our research specs. Taxonomical classification is totally different. It's focused on the fundamental biology and the evolutionary history of fungi. So what's the organizing principle here? Well, it's primarily based on how fungi reproduce sexually. And this brings us to a really critical point, the disconnect this system can create with the clinical world. See, taxonomic classification relies on observing the sexual state of a fungus, but in a culture you take from a patient, you almost never see that happen. So while a researcher might be obsessed with Ascomycota or Basidiomycota, a clinician might find the system kind of impractical for their daily work. And that last group, Deuteromycota, the imperfect fungi, that's basically a fascinating catch-all, a big mystery box for fungi where we just haven't observed a sexual state. At least, not yet. And hey, just to make this a little more memorable for you, think about this. The largest phylum, Ascomycota, isn't just home to common molds like Penicillium, it is also the group that contains the infamous cordyceps fungus, the so-called zombie fungus that's famous for taking over the bodies of insects. What a wild family member to have. All right, our third and final system is maybe the most direct one of all. It doesn't ask about disease, it doesn't ask about sexual cycles, it asks a much simpler question. What does it look like? This is classification based purely on morphology. It's physical form, and it's a system that can be used by pretty much anyone in any field. At its most basic level, we've got three main categories. You've got your yeasts, which are unicellular and reproduce by budding. Then you have molds, the filamentous fungi, which are made up of those long branching threads called hyphae. And then you have the really fascinating ones, the dimorphic fungi.
These guys exist as a mold out in the cool environment, and then bam, they transform into a yeast at warm body temperatures. It's a trait that's really common among many of the most significant pathogens.
Now, if we dig a little deeper into appearance, especially in the lab, there's another super useful distinction. Pigmentation. Is the fungus hyaline, meaning it lacks pigment and looks clear or colorless, or is it dimetaceous, containing melanin that gives it that distinct dark brown or even black color? This is such a quick visual cue that lets scientists communicate basic info while they're working on a full identification. And for those who want to get really granular with morphology, we can even look at how the asexual spores or conidia are formed. It's pretty cool. In thallic formation, an existing hyphal cell just kind of converts itself into a spore. But in blastic formation, a new spore actually buds off, growing out before it detaches. I mean, these subtle differences are absolutely key for making a precise microscopic ID.
So, we've explored 3 completely different lenses: the medical, the taxonomic, and the morphological. How in the world do we bring this all together? And the really big question is, which system should you be using?
This table right here, this is the key. This is the big aha moment. The crucial point is that these systems are not in conflict with each other. They're just different tools for answering different questions. If you're a clinician, you're asking, how does this affect my patient? You're going to use the medical system. If you're a researcher, you're probably asking, what is its evolutionary history? You'll use the taxonomic system. And if you're in the lab making an initial identification, you're asking the simplest question, what does it look like? And using the morphological system. And that is the ultimate takeaway. The best system is simply the one that answers your question. Let's think about Aspergillus for a second. From a medical perspective, it's an opportunistic pathogen. From a taxonomic perspective, it belongs to the phylum Ascomycota. And from a morphological perspective, it's a hyaline mold. All 3 of those statements are correct. All 3 are useful. They're just different tools in your mycological toolkit. Thank you so much for tuning in. So let me know in the comments, what is your favorite fungus? Or what's your favorite classification system? Which one do you use the most? The world of fungi is an amazing reminder that perspective is everything. The same organism can tell a completely different story depending on the question you ask. Tune in next time for more on this beautiful kingdom of organisms.